Thank you, everyone. It's great to be with you today, and thanks for taking the time to support Wisconsin Saltwise and some of these endeavors to not only understand ecological consequences of freshwater salinization and salt pollution, but ways of fixing the problem or addressing the problem, a role we all have. And so you probably heard on Monday, if you were there, Hillary Dugan was talking about um, how chloride concentrations or salt concentrations are increasing in our lake streams, wetlands, so on and so forth. So environmental concentrations are going up precipitously, and it's a wide variation, right? Some concentrations in lakes haven't changed all that much, some have gone up quite a bit. And so in the environment, there's a wide range of salt concentrations that now we want to know what are those ecological impacts. So if as I tell my ecology students, everything is interconnected. And so what my lab does, what my research does, is it focuses on once salt's in the water, what happens to the organism in freshwater ecosystems? How does it alter connections between predators and prey, fish and zooplankton, so on and so forth? And so when we think about freshwater ecosystems, or any ecosystem really, you can kind of think of it as this maybe random disorganized series of dominoes where they're not all in a line, but if you push one domino over, how many dominoes are going to fall? And so everything's connected. Aquatic terrestrial environments are connected. And so when we're dealing with this issue of salt pollution, or salts kind of getting into our freshwater ecosystems, what could happen ecologically? And really, that's the focus of my research, many of my collaborators' research. And so I'll share a little bit of that with you today. And I'll be happy to take your questions after we go through a few slides here. So when we do scientific research, or when we're looking at the issue of salt pollution, it's very useful to study things in the natural environment, understand what's going on in the natural environment, in our lakes, streams, and wetlands, and what salt concentrations exist there. And then, it's also, incumbent on us, we survey the biological communities, or the ecological communities. And so we've done a lot of that. And so we kind of get a handle of what's going on in a lake ecosystem, for example. And we do things called food web studies. So what you see here is a couple undergrads and myself. We surveyed this lake out in New York, and we looked at essentially the gut contents of the yellow perch population. So when you think about pollutants like salt, right, they can affect things like fish directly, or they can affect invertebrates directly, which may indirectly affect fish. So there's so many different connections in freshwater ecosystems that we really want to understand how they're going to change with this rise in salinity from overuse of road de-icing salts. So what we found in some of our work is that yellow perch, which I love eating yellow perch, a good perch fish fry on a Friday night or weekend or something like that, it's one of my favorite meals. In this lake in particular, and a lot of different lakes, this is probably true. Yellow perch love these things called amphipods. You might know them as scuds, or if you were go out into a stream or a wetland or even a lake, and you kind of take a handful of the bottom of the wetland or the lake or something like that, or stream, these are those things that kind of are just scurrying around in the mucky mess sometimes. But in a lot of ecosystems, they're a critically important food resource for things like yellow perch at certain times of the year. So from our field studies, we've identified certain relationships between organisms to really understand how salt pollution might affect them. Now, when we single out yellow perch and one of their main diet items, for instance, and we expose that diet item to a wide range of road salt concentrations, we can see a pretty noticeable decline in the abundance of some of these important food resources. And so, sometimes these impacts happen at high concentrations of salt, or low concentrations of salt, or moderate. Now, when I use those terms, low, moderate, high, they're all relative because they can mean different things to different people. But when we see high concentrations of salt, we see a pretty noticeable decline in these amphipod species. And so yellow perch love to feed on these amphipods. And what we're kind of working on modeling right now is that in a freshwater lake, for instance, if we were to reduce the availability of some of these critically important food resources, how might that affect certain fisheries? And so we want to scale up like a weather forecast, right? You take a bunch of information and you want to know what's going to happen in the future. So we can do that in ecology too. We can do experiments, we can do field work, understand pattern and process, and then model those things into the future. So in theory, if we see the decline in some of these important food resources, does that reduce food in a way that would lead to maybe slower growth or smaller adults, which in science, there's this concept of bioenergetics, right? It has the potential to produce fewer, smaller offspring. And so, as I mentioned before, everything's interconnected. So we're making these connections in natural environments, and then we're bringing those semi-natural environments into the lab to study how important resources like amphipods or fish food to some people might affect, the growth and abundance and health of different fish species. So I'm going to kind of run through a bunch of different trophic levels today. And so I'll talk about fish, I'll talk about other things. But in freshwater ecosystems, especially near the shoreline of lakes or in wetlands, we have a lot of macrophytes or important plants that generate pretty important, a series of habitat for organisms in lakes and wetlands. And so one of these that we've done some studies on is a freshwater charophyte. It's just a giant macroalgae. There's a bunch of macroalgae. There's a bunch of macrophytes in freshwater ecosystems. And you think about where you might like to fish for bass or shoreline fishes, right? It's in the weed bed, so to speak, which I would call macrophyte beds, and they're critically important habitats. But when we expose, this freshwater charophyte to different concentrations of salts, this being sodium chloride rock salt, we notice a pretty precipitous decline in the biomass of this filamentous algae. So it serves as not only refugia for fish and young fish and macroinvertebrates from other predators, but can also, provide a substrate and a food resource for a lot of invertebrate species. So we've noticed pretty precipitous declines as we increase our salt concentration in this freshwater macroalgae species. We've also done different kinds of experiments on fish, and so this is what you see here is rainbow trout. And so rainbow trout are a Pacific salmonid species, right? And so you think Pacific salmonid, Pacific salmon species of the genus Oncorhynchus for you fish heads out there. You would think that these are pretty tolerant to salt because, they go from freshwater streams of the Pacific Northwest, Northern Pacific Ocean, and they go out into the Pacific Ocean and back and forth again. And so we've introduced these all over the place in the United States. And we simply ask the question that if we expose what, is this salt tolerant species to different types of road de-icing salts at the early life stages, and we let them grow, in this case for 25 days, what might happen? And so really, you can go visit the paper if you'd like to look at the data, but really on the right, you can see in a high road salt concentration, we get substantially reduced growth just after 25 days. And so the larval fish in the middle is what we would call an alvin, right? In the salmonid life stages, it's yolk or sac fry. And so, they don't have the kidney function, the gill function, and a lot of different functions that they do as adults. And so when we expose them to high road salt concentrations, we see this noticeable reduction in growth. And how this might affect them long-term or later in life, we just don't know that yet. So, fish, invertebrates, the green stuff, the macrophytes, macroalgae in freshwater ecosystems affected by salt, okay? But one question we work on is whether current water quality guidelines really protect freshwater organisms and ecosystems. And so there's a couple of numbers I want you to remember for the next few minutes, and that's the chronic threshold of 120 and in the United States and Canada, 120 in Canada, and 230 in the United States. And so 120 milligrams of chloride per liter and 230 milligrams of chloride per liter. So keep these thresholds in mind. These are the chronic thresholds designed to protect the vast majority of freshwater organisms. And so what we can do are a series of experiments, and a lot of collaborators of mine, we all got together at a conference and said, let's take organisms from the natural environment, expose them to a wide range of salt concentrations, and let's ask how they respond. Is there a certain kind of geographical response that where some regions we should maybe be more concerned about salt pollutions than others? And so at each one of these sites you see here, a group of researchers went out, collected a natural zooplankton community. Maybe you're wondering, well, what are zooplankton? Zooplankton are one of the most important organisms in freshwater ecosystems. You don't get fish without zooplankton. Zooplankton consume algae, phytoplankton, things like that. And then they grow and reproduce, and then all fish species at the early life stages will consume zooplankton, so they are able to grow and maybe become predators or eat zooplankton throughout the rest of their lives. So they really take all that green biomass in freshwater ecosystems, transform a lot of that into their own, and allow higher trophic levels, the fish, the things we might enjoy angling for, to thrive and grow. So at each one of these sites, we took a natural zooplankton community from the lake, exposed it to a wide range of salt concentrations, and I'll just kind of, for the sake of time, get into what we found. Now, what you see here, just to orient you a little bit, on the y-axis is just the percent change in abundance, and on the x-axis is the study site you just saw on the previous slide. So this is a group of cladocerans. Cladocerans are just kind of large body zooplankton. You can see them with the naked eye, but they're pretty ubiquitous across freshwater ecosystems worldwide, and in a lot of the lakes and wetlands. So if you look at the dashed line, that's if it's above the dashed line, it's an increase in their abundance. If it's below the dashed line, it's a decrease in their abundance. And I think if you look at the vast majority of the study sites, they're all below that dashed line, and then the solid red line is a 50% decline line. Now, each dot is the Canadian threshold of 120. Remember, I told you to remember these thresholds. And the kind of gold color one is the US threshold of 230 milligrams of chloride per liter. And what we see is that at these concentrations, the vast majority of cladoceran communities in lakes that we studied decline quite a bit at those threshold concentrations from a control concentration which has no salt in it. Okay, we can look at this a little different way. We can ask the question, at what point does 50% of the cladocerans, at what point does 50% of their abundance decline relative to our no salt control concentration? And so again, the red dashed line being the US threshold, the solid line being the Canadian threshold, and we can see the dot being the average response, and then the 95% confidence interval. And so anything that's red, its dot or its confidence interval overlap with those thresholds concentrations, indicating that already at those concentrations, 50% of the biomass was already lost. Okay, so that's a substantial reduction, and kind of indicates that once we reach these threshold concentrations already in many lake ecosystems, we're already losing quite a few organisms, and I would argue important organisms at that. So when you lose that grazing pressure, you lose the things that eat the algae in freshwater ecosystems, you get more algae. You've lost grazing pressure. And so at about half of our study sites, these are not tanks from that specific experiment, but the change from the left to the right tank is that change is exactly what we saw in that experiment at some of the study sites. So the magnitude is relatively the same. And so we lose zooplankton. We see this kind of clear water phase on the left. And as we move to the right, we kind of see an increase in the green stuff, which is important, but we get more green stuff in the water. And so we would call this in ecology a cascading effect. And so, yeah, so it becomes a question, do these things really protect freshwater organisms? And I would suggest not all of them. we've seen pretty substantial declines in this study of, freshwater zooplankton and a concomitant increase in algae. So some of us, we're just like, why do these differ from some of the science that's already out there, some of the laboratory tests that's been out there? Well, in our experiment, we used wild strains. We also had in our experiment species interaction. So it was a semi-natural ecological community. And when we think about some tests that are done, they use classic, for the sake of a better term, the lab rats, right? There are model organisms that we use in science because a lot of science has been done. We know them well and how they should or shouldn't respond to certain things. So in addition to not using target species of only the lab rats, there are several lakes that are really dilute in, they're really clear water lakes, thinking about the Canadian Shield. They have low concentrations of different solutes, low concentrations of food. And so they may respond very differently than a laboratory test that has perfect conditions, overabundance of food, no predators, no competitors, so on and so forth. So we need to think about, that natural environment and those interactions when we conduct these tests. And then, just one other thing to think about is, what about everything else in the water? So what I've shown you here is just about salt, right? But we know there are PFAS, microplastics, invasive species, other pollutants in freshwater ecosystems. And so, that's one question that we also work on, is the interactive effects of different, what we would call stressors. Our group, a lot of research groups are finding that low concentrations of salt can trigger pretty substantial ecological changes. And I'd like to give a shout out to Dr. Shelley Arnott's group at Queen's University in Canada, who's come out with a great series of papers showing that at concentrations ranging from about 4 to 40 milligrams of chloride per liter, you can see a pretty noticeable change in zooplankton communities. And those are really low concentrations. So it is a bit alarming, no doubt. And so I think we can think of other studies, from Hillary's work or Hillary's discussion on Monday. You probably know some lakes are, 5 milligrams of chloride per liter, some are 50, some are 100, so on and so forth. And so kind of keep her talk in mind when we think about these ecological impacts, because another paper out of the same group at Queen's, finds that the LC50s and so on and so forth are pretty low, 23 and 60 milligrams of chloride per liter. Remember, those thresholds currently published by governmental agencies are 120, 230, so on and so forth. So I think, it's time to rethink how salinity and salts affect freshwater organisms. And it's not all about just survival, death, changes in biomass and abundance. Some fish species they're really tolerant to salt, right? I mean, you can put thousands of milligrams of chloride per liter into the water and, they may not die. You may or may not see reductions in growth, but there's also this sublethal behavioral avoidance. And one paper I really like is from Ray Morgan, that shows at really low chloride concentrations in streams, 33 to 108 milligrams per liter, it can change fish assemblages. And it's not due to a reduction in growth. It's not due to death, but it's just behavioral avoidance. And so the salty plumes cause some fish to leave certain stream reaches, and so I think there are behavioral changes that occur in some organisms, just avoidance of the salty plumes that has implications for ecological impacts. So when we try to summarize a lot of this stuff and we try to synthesize, well, what are the possibilities for, in this instance, lake ecosystems? We see, Allison mentioned my sister's husband, he loves to fish for muskie in lakes all around Wisconsin and Minnesota. And so that red X you see on the left, if we knock out zooplankton communities at certain concentrations in lakes, if lakes get contaminated to some certain extent, we need to think about those connections among the trophic levels. So we lose our zooplankton. Maybe we get a little more algae. Will the planktivorous fishes or young fishes have the right amount of food and the right kinds of food to eat? That's a question we need to address. And how does that scale up to even some of the largest fishes in some lakes? And so, moving back to the right, we think about salt pollution, reductions in zooplankton diversity and abundance, enhanced algal biomass, reduced invertebrate biomass, the critters on the bottom of the lakes, perhaps a reduction in some macrophyte species or some macroalgae species. And how will that change our freshwater ecosystems? And I'm not sure we want to really get there to know, but there's a lot more work to be done, but do we really want to find that out? So local solutions to big problems are, one way of doing this. And Wisconsin Saltwise, I think, is the, one of the ultimate organizations doing this. And we're doing this in Northwestern Ohio too, because if you look at all of these dots, I don't mean to go over my time here, but each one of these dots, next to each one of these dots, is the chloride concentration for a stream in Northwestern Ohio that all flow to the Maumee River, the largest Great Lakes watershed in the Great Lakes, and to Lake Erie. So most of these values should probably be less than 6. Geologically speaking, for this area, the salt deposits are all north and east of us. So surface stream water concentrations here should probably be less than about 6 milligrams per liter. But you can see these numbers are far above that. So when you go to different areas, this picture is probably pretty common. And so that's all I have. I apologize for going a little bit over time, but happy to take any questions, or if you want to email me, I have my email here. So thank you very much.
Thank you so much, Bill. It was excellent. I realized that I forgot to tell people to type questions into the chat. So I will do that now. And I'm also going to type my email into the chat, wisaltwise@gmail.com. There was someone yesterday who said they had some issues with the chat. So that's always an option as well. So hoping that we get a few questions from folks in one of those ways or another. Let me just—
I apologize if my voice was a little hoarse. I had lecture right before this, so I was boring my students for a while.
You're good. I'm gonna end your sharing. There we go. So we both pop up on here, but can see our faces again. All right. So let me keep an eye. All right. We do have some questions that have come in, Bill. The first one from Travis. Why did cladocerans increase in some of the studies? Were there any other zooplankton that were reduced, which allowed cladocerans to flourish?
Yeah, so there's probably some turnover in the species of cladocerans in different studies. So when you move across locations, some of our study sites, there could have been, genetic strains that were tolerant to salt. And so when you lose maybe some of the competitors, this is our prevailing hypothesis, worth testing. But when you lose certain species, other species competitive with least can allow other species to thrive, right? And so they're tolerant. And so you can see an increase in the abundance of certain species, which kind of, for the entire group, might increase that biomass even more, right? And so, different locations around the world have different genetic strains that have different evolutionary histories. And so if there's an evolutionary history with high salinity, or they're always exposed to high salinity, there might be some resistance or resilience there in the community to where certain species thrive under salt, but others have declined. And so that could be one reason maybe a sensitive species just kind of shot through the roof, for lack of a better term.
All right, thank you. All right, then we've got a question from Paul. Are aquatic invasive species able to withstand higher salt concentrations? Would this have any effect on long-term on river lake ecosystems, perhaps increasing their negative effects to higher levels? So I guess speaking broadly, aquatic invasive species would we expect them to be able to potentially withstand higher salt concentrations? And then what would be the long-term effects of that on the ecosystem?
Yeah. Some can, some can't, right? So there's been some scientific studies that have shown some freshwater invasive species are highly tolerant to increases in salinity. So some are not. We have some experimental work, some experimental evidence that needs to be published yet. So I don't like speaking to that all of the time. I like the scientific community to evaluate it first. But it's some invasive species are tolerant and some aren't. And so it's all going to depend on the functional role of a given invasive species, how they might affect in the ecosystem. So different scenarios. An invasive species is highly tolerant to salt. It thrives in a high salty environment. You would expect the magnitude of its impacts to increase as other sensitive native species decline. And so, I think there's a lot more area or a lot more research that can be done in this area. So, I think it depends. And so it's just which invasive species are we talking about? Has the science caught up? But yeah. So you might expect, like, I don't know, if zebra mussels tolerant to salt, right? Their clearing capacity of algae out of the water column might be magnified if other bivalves or filter feeders are susceptible to salt. So yeah, very few studies on that, but I'd like to see a lot more.
One of the first things that comes to my mind with salt tolerant species are Phragmites, right? So
Oh yeah,
those giant, kind of what people might think of, I don't know necessarily giant cattails, but giant plants growing in roadside environments.
yeah.
Sometimes to the point that it maybe even occludes your visibility. Uh,
Yeah.
but yeah, Brandon has a question here. What could the average person without training do to see if a water body is affected by road salt? Lab tests, visuals.
Yeah. You could probably go on some online shopping stores that you could get a salinity meter. They're really cheap. So for fish tanks, you could get a salinity meter and you can either dip it in the water or get a cup of water and dip the meter in there. They range anywhere from think like 15 to 35 bucks. And so, if you want to know what any water body, what the salinity is, salinity meters are pretty cheap. And so you can pretty much get them at any box store or online stores, things like that. And you can go out, measure your wetland or lake or whatever you want. And if you want to do the back calculations to figure out how much chloride is in there, you have to first assume that it's sodium chloride. So that's a little bit tougher to figure out. But there are a lot of cheap meters out there that can be purchased by anybody to measure the salinity of a freshwater body.
I also just put a link into the Isaac Walton League. I think this question would come up maybe on Monday with Hillary's talk. The Isaac Walton League provides, I think it's like 4 free test strips to any resident. Just say, hey, I want to test a water body. You can also buy those test strips online as well.
Yeah.
Cheaper than lab tests. I don't know. Kind of the salinity meter might be cheaper than a whole bottle of the test strips. They aren't. Yeah, I don't know. They're like $1 or $2 each.
Yeah,
But pretty
I
easy
think,
to use.
yeah. For 20 bucks, you can get something pretty cheap. Yeah.
Brandon had another question here. Actually, maybe a few are coming in. I'm trying to read through what species, whether fish food or fish, are most susceptible to chlorides.
I think the vast majority of the work's been done on zooplankton, right? And so there's so much room for expansion, and we just don't know. There's some classic species that are used in studies, and I think what's clear from some of our work and from a whole host of work, I mentioned Shelley Arnott from Queens, is that zooplankton are kind of this important link in food webs in freshwater ecosystems, lakes and wetlands, and so they're probably the most susceptible, but we don't even know enough about freshwater algal species, right? We don't know enough about bacterial communities in freshwater ecosystems. How susceptible are the bacteria that are transforming nutrients? How susceptible are they? And so, yeah, I would say starting with zooplankton, but I would also look at smaller organisms, bacteria, um, phytoplankton, things like that, that we really need some research on. So
Yeah, I do know there is some cool research on bacterial communities that came out of University of Wisconsin Milwaukee,
sure.
because
Yeah.
we've taken some of that and actually packaged it up for high school students, thanks to the work of Lexi Pasante, who did the work and then created some lesson plans through the Data Nugget program. But just a little plug for that.
Yeah, no.
um,
Yeah,
so yeah, I want to take a quick pause here. I do know we're at that 1 o'clock or just past that 1 o'clock time. So if folks do need to pop off, totally get it. I think Bill, if you've got a minute, I'll ask you a couple more questions.
sure.
Um, but yeah, otherwise we hope that people can join on later to catch what you might miss and hopefully see us again tomorrow. So looks like we got a couple more questions. Brandon has said again, okay, Brandon's really getting the questions out there. Thank you, Brandon. You briefly mentioned other stressors besides chlorides. So stressors like microplastics, PFAS. Have you found any other parameters that are synergistic, making chloride more toxic at lower levels?
Yeah, I mean, so there's natural and anthropogenic stressors, and so some people have looked at temperature, for instance, that as we warm the waters of freshwater ecosystems, the surface waters, warming temperatures can interact with salts to have a greater negative effect, right? So effects can be additive, synergistic, so on and so forth. And so what we've found is like even natural stressors like the fear of fish predation, for instance, can magnify the effect that salt has. There's an interactive effect there. So when things are scared in the environment, that that response to predators with salt toxicity, with salt effects can magnify responses, right? And so yeah, there's a lot we don't know about interactive effects, but in a lot of ecological studies, it's showing that, I think, warming water temperature, stress with, natural predators and things like that can interact with salt. So we do need to consider, there's so many different concurrent stressors in freshwater ecosystems that piecing that together is really notoriously difficult in a scientific way, but a lot of people are working on it. So the answer is yes. And which ones? I've seen temperature, predation, things like that. Microplastics, no one, or not that I know of. But yeah, so room for a lot of work.
So Bill will continue to be needing new masters and PhD students, is what I'm hearing.
Yeah, more funding too.
And more funding. All right. So it looks like our last comment here from Ashley, just saying that mayfly larvae is pretty susceptible. I think that was in response to one of the earlier questions, but yeah, that's what I've got. It looks like we maybe have an email or two here, Bill. People want to be in touch with you, so I will be passing your email along.
Sure.
But thank you so much for joining us today and sharing your work, ongoing work. And yeah, we'll look forward to the newest research when it gets published and otherwise ongoing conversations around policy as well. So I had shared in the email, one of the things I really admire about you is that you're not just in the ivory tower doing the science, you're really actively getting out and trying to make positive change in this world. So thank you for all of that.
Yes. Well, thank you, Allison. I appreciate the venue and thanks for all the efforts with Wisconsin Saltwise. I think, everyone who's here, we all have a role to play. And I think it's pretty groovy that everyone is kind of, people are starting to pay attention. It's awesome. So cool.
All right. Well,
Thanks,
take care,
everyone.
Bill, and yeah, see you next time.
All right. Bye.